So DNA, I'm just going to repeat this really quick for the camera. So DNA codes for mRNA, mRNA codes for tRNA, which brings in amino acid, proteins, traits. If we change the sequence of amino acids, we change the shape of the protein, which changes our function of our proteins, which then in effect can change the traits. Okay, back to where I was. COVID. It's a virus. Viruses are non-living things, but they do contain genetic information. They do contain genetic information. The genetic information found in COVID is in the RNA sequence. So they don't have DNA, they have RNA. Non-living thing, but they have RNA. They have some coded messages in their genetic sequence. They are not able to self-replicate. And we talked about this a little bit when we talked about cells. COVID will actually, once inserted into your body, into your cells, once you get it into your body, it'll inject its DNA, or RNA, I'm sorry, its RNA into your cells. And then it hijacks your ribosomes to do this process. Okay? So COVID is RNA. So its RNA sequence on our ribosomes will go through and assemble amino acids, which then make proteins. And really the proteins that they're making is more COVID. Okay? So COVID inserts its RNA into our body, which then goes into our cells to make more COVID. And then what happens to those COVIDs? They leave our cells, they go to new cells in our body, process is repeated, you get infected throughout your entire body. And that's how you get one, multiplies many, many times. COVID. It has genetic information. It uses this exact same process in our bodies, in our cells, to do this. Okay, now we do the similar things when we go and get vaccinated. Now, well, actually, before we get vaccinated, let's talk about COVID. If you get COVID, okay, you get COVID, your body recognizes as a natural defense mechanism certain structures of the infection, and you build these things called proteins that are specifically called antibodies. So if you got COVID, you have antibodies in your body. And those antibodies are there to try to prevent you from getting infected again. Okay, so your body's natural resistance or natural response. It's your body's natural response to having COVID is you will make these proteins called antibodies that help fight future infections. If you were to go and get a vaccination, there's 3 different vaccinations in the United States. There's the Johnson & Johnson vaccination, there's the Moderna vaccination, and there's the Pfizer vaccination. Now, Johnson & Johnson used a process, a technique that used, I believe it's called an endovirus. It used another virus to insert a molecular sequence to produce proteins. Pfizer and Moderna were the first time commercially we've ever done this type of vaccination. They've been testing them for years. It's just the first time we've actually mass produced them to use. Is the vaccination of Pfizer and Moderna were mRNA. So if you were to go and get a Pfizer or Moderna vaccination, they injected you with a vaccine which is in the form of mRNA. It's very similar to what the virus did, is it takes the it inserted its genetic information to use your cells to produce more COVID. It's similar, but the mRNA that they injected into you wasn't make COVID. The mRNA that they give you in the vaccination goes through this process to assemble amino acids which make antibodies. So instead of COVID making COVID, this vaccine makes the antibodies, which then changes the shape of the spike proteins on COVID, which then should change how they function in terms of how they enter cells. That's that idea. The cool thing about an mRNA vaccination is it's kind of sorta, like sitting down at a keyboard and saying, what letters do I want? Because we can put these sequences in a pretty much any order we want, and we could say, well, I need a start codon, so I need an AUG. What do we want next? What amino acid do we want to come next? Well, I want a leucine, so that will be a da-da-da. And then I want guanine, no, not guanine, that's a base pair. Tryptophan, da-da-da-da, okay. And they can basically go through and they can type, not really type, but they can go through and choose what they want the mRNA sequence to be. It's not like, well, oh, let's see which one's going to work best. We can go through and say, this is the sequence I want. And then once they can fairly easily mass produce these, and then that idea is then we can make the antibody that we want, which has some neat potential, because what happens if something changes? Well, we can just tweak the sequence we have. Well, now it's not a leucine tryptophan, now it's a leucine valine. And we should be relatively easily able to flip and insert a new amino acid to meet the change that is occurring. So it's a newer style it's a new delivery technique is really what it was. But again, been tested for about a decade prior to actually being used in this vaccination process. So when we talk about COVID, December-ish, 2019 is when the original strand was kind of talked about and was kind of discovered. Actually, let me take a step back. There's some people that are concerned that, well, if they inject mRNA into my body, isn't that going to be part of my DNA forever? And what the scientists say is, the answer is no, because it goes from DNA to mRNA, not mRNA to DNA, right? It's a flow of traits this way. You have mRNA in your body because you have DNA that codes for the mRNA, which then go da-da-da-da. What happens to your mRNA that say make blue eyes? Well, once it's done making those blue eye proteins, eventually they get chopped back up into nucleotides floating around. Oh, now I want to insert new protein, and they get it reassembled. The same idea happens with the mRNA vaccination, is at some point in your body, and it was a couple of weeks, I think, window, that mRNA has already been chopped up and it's gone, from what I have read. So it's not a forever in your body type of thing. But this idea of COVID went through and made more COVID. When we talked about COVID, December 2019, that was like what we considered to be like the original strand. But then we had this thing called Delta, and then there was that Omicron thing. That was only like not too long ago. What would they call those? Those are different blank of COVID, different variants. And what is a variant? What is that? It's a mutation. That's all it is. Is during this time frame, instead of COVID making more COVID at some point in this process, oops, mutation, it still made more COVID, but it made a oops version of COVID. It's a mutation. And what does a mutation do? Well, sometimes it's good, sometimes it's bad, and sometimes it's whatever. Some of these variants, there's more than just those two variants out there, by the way, that we talked about, and they were just the ones that kind of spread throughout the global world. But some of those variants actually made it less infectious. So if it's less infectious, it's less likely to spread, it was less likely to make it around the world, which that's why we didn't talk about it very much. Some of these oopsies made them more infectious, which is why Delta became the dominant strand over the summer, because it was more infectious than the original strand. It was more likely to get spread. It was easier to get spread. But that's all they are, is mutations. But anytime this process is going on, there's a possibility of mistakes. So the less times that COVID gets replicated, the less likely a mistake is going to get made. Just kind of keep that in mind. Every time we copy something, there's a potential for mistakes. The less time we try to make any copy of anything, we're better off in terms of less likely to make those mistakes. So this is kind of the natural process. Yesterday, right at the end of class, I showed a video, and the video talked about CRISPR. And CRISPR is a way that we, as humans, can go through and we can change this sequence. We can manipulate, we can choose what to change. And if we change this, we can change this. So if we change our DNA sequence, we can change the traits being produced. It's kind of new, 5 to 10-ish years, I'd say, it's somewhere in that ballpark. We're getting better at it. We're not great at it, but we're getting better at it. It's pretty precise. We can go through and we can find a very specific sequence. We can cut that bad sequence out, and we can put in basically whatever we want. So when and can and what should we do with it? So I'm going to throw some situations out there just to kind of think about it. Is that okay? Is that not okay? Is that good? Is it not good? So what if you had you were going to have a kid. Let's say you were going to have a kid, and you want your kid to have a certain eye color, or you want them to have a certain hair color, or you want them to have a certain athletic build. You want them to be male versus female. You want them to be above average intelligence. Is that okay? Is that okay for us? Because, by the way, it's doable with this technique. It could be doable. Is that okay for us to go through and say, well, let's change that. I want my kid to look like this. Is that okay? And this is just kind of for you to think and not really necessarily respond to. But is that okay? I want my kid to be above average intelligence. I want them to be smarter than everyone else in the room because I think intelligence is going to lead to success. Is that okay? I want my kid to be 6'6", and athletic, with great hand-eye coordination and a good muscular build. Why? I want them to be a D1 scholar athlete so they can get a full ride to some university, maybe even go pro. Is that okay? Is it okay for me to choose that before I have my baby? Or not? And I'm thinking a lot of you are saying, well, no. Okay, fine. I get it. But we're not too far from probably the technology that we could start making some of these choices. They refer to in the video as designer babies. And this idea is we can kind of customize what do we want. Okay, so let's get away from that because, okay, well that's maybe not what most people would say is good or okay. But what if, let's just say you are a diabetic, okay, and you do not produce insulin. Your body doesn't produce insulin. So because of that, you're on an insulin pump, you're careful with your diet, because something happened in your DNA sequence that doesn't say make insulin. What if we find that part in your DNA that says make insulin, we fix it, we delete that bad part, we put the new part in, and now you're making insulin. You're no longer a diabetic. Is that okay? What if you had cancer, or a loved one had cancer, and we could delete that section that says, hey, make these irregular cancer cells? Should we? Could we? Can we? Do it? Okay. What if I am 80-some years old, and hey, there's something in your DNA that says, this isn't good. Could I fix it? What if I'm 40 years old? What if I'm 5 years old? What if I'm not born yet? At what point is it okay to say yes versus no? Who makes that decision? Is it okay for you as a parent to say, this is what I want my 5-year-old to have because I think it's what's best? Or do you have to be a age of consent? So you gotta be 18 years old before you make that decision for yourself. Okay, these are things that we're gonna have to start answering questions as a society fairly soon. They referenced it yesterday. There's some ethical issues, and they kind of draw a line. They say they draw a line in the sand, which basically means like, here's the line, don't step over it. Okay? And there are certain conditions that when we talk about humans, that what's okay and what's not okay to do with this DNA sequencing or editing. And originally, there was a couple criteria. The two that I remember is consent. You have to be the consenting individual, and it can only affect you. It can only change you. You say, well, who else would it change? If I change some cells in my arm, who does that change? It changes my DNA in my arms, right? Well, what if I change it in my heart? Well, it changes in my heart. What if I change it at a younger age and it affects my body cells? Well, now it's affecting me. But if I were to mutate or change the DNA sequence of, say, sex cells, your sperm cells, that doesn't impact me or just me, it impacts the next generation, and the next generation, and the next generation. Okay? And that's where they said, no, it can only impact you, there's certain, like, you have to have consent. It's been a couple years now, there was a Chinese scientist who did some research, and he went through and he edited the sequence of some unborn children. And he went through, and they're often referred to as twins, as the twins, the Chinese twins, CRISPR twins. And he went through and he edited, and the line is referred to as a germ line cell. So he edited the DNA sequence of these children at a really, really, really early stage, like a couple cells old stage. And then what happened is, as those fertilized eggs then developed, that genetic sequence that he changed really early on changed every cell in their body. They didn't consent to it, they're unborn. Mom, I think, was the one who consented. But it changes their sex cells, which means that their children's will have these altered man-made genes, and the next generation, their children's children and children's children's children. And that was, no, that's unethical. But he did it anyways. I just skimmed a headline, 2020, January 2020, he was arrested and put in jail for 3 years. The judge ruled for 3 years because he was illegally practicing medicine. He crossed these ethical lines, and he did it. Now, why did he do it? From what I have read, it's been a little bit since I reread some of these articles. Originally, though, his rationale for doing this was mom was HIV, I believe mom was HIV negative, dad was HIV positive. So what he altered, he edited, was the infection, was to reduce the likelihood of HIV infection. That was the intent, that's what he claims the intent of the editing is. So that children would be less likely to get HIV. Now, from what I've read, the likelihood of children getting HIV because dad is HIV isn't super high. So there's some arguments that they were low risk to begin with, but that's what he said for HIV resistance. So you can say, well, as a world population, wouldn't that be a good thing to make people more resistant to HIV? By the way, there's some natural mutations that occur that currently have people immune to HIV. There's probably people out there that have a mutation that makes them immune to COVID, that no matter what, they won't get COVID because of some random mutation they have. One of the potential side effects of his editing, though, is in other species where they did that editing, is above average intelligence. So these twins that got edited their DNA because of HIV resistance potentially have an above average intelligence because of the sequence that he had. Now, was that the intent? Was that truly a side effect? I don't know. But it's one of those things about what's okay and what's not okay. And again, as a society, these are some choices that we, as a society, are going to have to kind of have these conversations and figure out, because our technology is not too far off for some of these things to potentially happen. Okay. So, for the rest of the class, I have an activity for you. It's similar to the activity that you did yesterday. It's looking at mutations, but instead of looking at 2 days ago. Instead of looking at mutations in the form of words in a statement or words in a sentence, it's looking at amino acid sequences. So when we change our DNA, we change the amino acids, which then in fact change traits. So this is looking at amino acids specifically. I'm giving you a half sheet which has an mRNA codon chart, and the idea is for you to kind of hang on to this, and now you have one. We're going to use it again on Monday in a different activity. One thing to note on this red sheet is there is no start codon, but at about 12 o'clock on your chart, the sequence AUG is listed. And AUG codes for the amino acid methionine. I can't ever pronounce it. But it's MET, is a 3-letter abbreviation. This AUG is a start codon, and it's located, AUG is located like right here, right around 12 o'clock. AUG is a start codon. It doesn't say start on this chart, but it is a start. So all of these do have a start codon. AUG is MET. That is our start sequence. That is our start codon. So this activity is due in Canvas. A lot of you are probably going to have it done before you walk out of here today. It's due on Tuesday at 11:59. Juniors, you are taking the ACT on Tuesday. Sophomores and seniors, no school. Next Thursday, we as 5-6 don't have class because sophomores will be at Career Fest. So just so you're aware of that. Okay, so this is due in Canvas on Tuesday at 11:59. A lot of people have finished this before they walked out of here today. The questions are similar to yesterday's, but yesterday's were more like identify the differences in the DNA sequence, where now today it's what is the type of mutation, using your notes. We're talking frame shift or point mutations is what we're looking for. So as you work through this, if you have questions, make sure you ask. I'd be happy to help you out with any of this.